Okay, it's working. Oh, this has a delay.
Oh, look at you.
Okay, it's oh, this has a delay. Guys, I'm really cute from the
It's
side.
like multi-minute delay.
Yeah, it's live. Yeah, it's live. Okay, so I need to turn off the. Okay, I tried to turn off the one where we could hear ourselves, and instead I turned off the one that's streaming. No. No, I just meant like. We should quit other things.
But it's like about hybridization.
What's happening?
What is that? Hey, you want to go? Good. Good. I must have recognized you. No, because Good. Yeah. my hair for a long time, which I think tells me how I am. Just like a long time. How's Boston? Yeah. Yeah, it's good. I've been gone more than I've been there, so yeah. Hopefully people keep asking me for like, but no, it's good. Yeah, I mean, it's like a weird distinction, right? It's like weird to be back. I'm like, oh yeah, I never left, you know, like, you can just take it. Come on. Yeah, I know. It's a ways. I got 8 a.m. class. Not me. Not Molly. Thank you.
We didn't actually go live, just.
No, we're live. I just want to make sure it's gonna work. I turned the sound off though, so it should be live. and
Perfect,
People
so
can
it's
now
just
see
going.
you, but not
Oh,
hear.
perfect.
And I'm telling you all
Alright.
of this information.
Perfect, because
Say what you speak.
okay, then you're good.
Say what you speak.
But Scott wants to sit in the back row.
Oh, well, Scott can sit wherever you want. You can sit wherever you want.
We go on the other side.
Yeah. I did the same thing.
Yeah. I come around.
We're gonna do ourselves.
Sitting next to me won't be good. And the empanadas, what was your final decision?
I baked them, but
Margaret, good to see you.
Sorry.
3 minutes, 5 minutes ago.
And I have a hamble in the foil if you want to take it. Hello.
How's it going?
Good.
Remember Silke?
Oh, hello. Yes, of course. I do remember you.
I
Good to see you.
This is my friend.
I love the dance.
This was the requested.
Oh, I see.
Mother's room. Wir waren beim Essen mal hier aber ihr braucht nicht mehr die Yeah. And we have I think so.
Yes, it's really nice. Oh, you want me to call me kind of talk about this? That's all. Yeah, Molly also. I know. She's doing at least.
Stacey, remember Stacey? She drew that. Stacey used to leave us notes, like she would draw things and sit down for Molly and I on our desk. We all shared an office. And this one she left Molly at some point. And then Molly digitized it.
Like the resources. Like
She's a grad student in Michigan, but not in Michigan where the other Sarahs are, at the other universities. All the Sarahs moved to Michigan. It's like there's a mass exodus of everyone named Sarah to Michigan. Yeah, she works on squirrels. Erin found her right. She's right there.
I think there's still a lot there. State of
I think they're rolling in now.
Scott found it.
I should be God about it. He's been a brainer.
Hello.
He needs help.
We can just. My lectures.
You might be surprised.
Yeah. So,
yeah. Molly looks good. Everybody looks good. I'm so excited. I thought I might get nervous, but I feel awesome.
I know. I actually don't feel that nervous.
Am I smiling?
I just more feel like this isn't actually happening. Wait, so you said the sound's not on, or this is all being recorded for posterity?
I was so nervous. At 9 in the morning, she was like, "Whoa!" And I was like, "Dude."
Okay, everyone. Let's get started on Molly Womack's dissertation defense. Molly came to us from University of Florida, and we were just so lucky that somehow we found each other. Molly has been really productive. She started up this system she's going to talk about today. She helped me get it funded by working with me on 2 NSF grants, and she's really made amazing progress. And any of you attended last year's award ceremony know that she's been rewarded many times with, for example, Department Teaching and Mentoring Award, DDIG, NSF Postdoctoral Fellowship, and more. But really what's been so fabulous about working with Molly is that she's just a pleasure to have around, and she's a real leader in both the lab and the department. And part of that leadership and productivity really comes from her independence, because Molly sees what needs to be done and she just does it. Actually, one of her letter writers wrote in her recommendation letters to me that Molly was perhaps too independent. And when I read that, I thought, what's too independent? Of course, there's no such thing. But then, maybe in her second year in the lab, I'm not sure, she was planning the first big field trip to Ecuador with me. And I thought back on that and thought maybe Molly is too independent. Mainly because she wasn't coming in my office all the time asking me questions. And I was like, is she even planning this? I don't know. But instead of Molly changing, what happened was I just came to realize that she's extremely competent, and I just could trust that when she had a question, she would come and ask me about it. And that otherwise, I should just not worry about things. So that's going really well. It's been really fun. And another thing that's been really fun is when Molly interviewed, she said she wanted to join the Hope Lab because she was really interested in biology very broadly, and she wasn't sure what she wanted to focus on. And she was pretty confident that being in the Hope Lab would give her a lot of exposure to lots of different parts of biology. And as you'll be about to see, she not only has had an exposure to lots of different fields of biology, she's really mastered it because she's incorporated into her thesis such a diversity of methods that she's done with such expertise. This talk is just going to represent a subset of what she's done. And as you'll also see, she's really figured out what she is passionate about. This is something that everyone on her committee, I think, was thinking, some committee meeting, Molly will be going through the doldrums just like a normal graduate student, and she won't be so excited. But it never happened. She really can't wait until this defense is over so she can start on the 3 new projects that she's planning on finishing in the next few weeks. So So this makes me really hopeful that this, what you're going to see today, is really just the beginning, and that we're going to collaborate for a long time, and we're going to see lots more really beautiful work from Molly. With that, I'll turn it over to her so she can tell you about the work.
Thank you, Kim. That was a really nice introduction. Whew. I have a lot of things to say about Kim as well, but I'm going to leave that to the end. And first tell you about the research that I've been doing for my PhD over the last 6 years here at CSU. So before I dive right into earless toads, I first want to start with the diversity slide that most evolutionary biologists have in their talk. And the reason we all have this is because on some basic level as evolutionary biologists, we're all interested in how and why this crazy diversity of life that we see around us has come to be. And in studying how evolution has shaped that diversity of life, it can be really informative not to just look at creatures that look very different, such as on this slide, but creatures that look the same. So convergent evolution occurs when many species evolve the same or similar traits. So we see this in two examples here, in burrowing mammals on the left, and in aquatic animals on the right. Now, largely, convergent evolution is attributed to shared selection pressures on these different species. These are pretty obvious in these examples. So the direct selection pressure to be able to dig and move through the ground, and the selection pressure to move efficiently through water. However, there are other things which could contribute to a trait evolving again and again in different species. And this is a great example of this comes in eye loss in cavefish. So cavefish are these cool organisms that have evolved a number of convergent traits. So you can see here that they lack pigment, but they also have highly reduced and often completely absent eyes. And for a long time, people thought that this was due to the fact that they were in cave environments, so they have this shared selection pressure that makes the eye less necessary. There's not a lot of light, so the eye is less useful. And also the eye has a high metabolic cost. And these two things were thought to drive this convergence. However, there's another big player. So what I'm showing you here is a cavefish face, not a bulldog. And that is what you see on the really bright dots there at the front of the mouth are the taste buds. And taste buds are enhanced in fish that lack eyes. And that's because these two traits, eyes and taste buds, are developmentally linked, such that when you reduce the eye, you get enhanced taste buds. And you really can't do one without the other, it seems. So this developmental link is really helping drive the convergence across these cavefish. And this really points out that we need to be taking into account development when we think about the evolutionary trajectory of a species. So to more broadly illustrate why, I'm going to give you a cartoon example. So we have some individuals here, these red dots, and they're found in a habitat, which is this yellow box. And in this scenario, we're going to consider color matching to be adaptive, such that ideally the dots match the box. Now we know random mutations are always occurring in populations, and this generates phenotypic variations or variation in traits. That's shown by these different colors in the second box. And selection can then act on this phenotypic variation and drive adaptive phenotypes such as the yellow dot to fixation. Now what we appreciate with developmental bias is that yes, random mutations are occurring, but because development has a hand in how those mutations turn into phenotypes, the phenotypes that arise are not always random. And some traits might be more likely to occur than others from the same or different mutations. I'm showing this in an exaggerated way by having this tan phenotype occur again and again in the second box. Now, selection can only act on phenotypic variation that is generated, and so in this way, it's really the combination of what we call developmental bias and selection that shape the evolutionary trajectory of a species. So in this way, it's easy to see how developmental bias can contribute to convergent evolution. If you have two species that develop in similar ways, they might have a similar developmental bias, and thus certain traits are going to be more likely to arise from mutation in both populations, and they're more likely to converge on those phenotypes. However, studying developmental bias is really hard. It's hard to find a system to really get at developmental bias, mainly because convergent systems would be great, but you usually have these strong direct selection pressures acting on those traits. And when you have strong direct selection pressure acting on a trait, trying to figure out whether developmental bias is influencing and how much it's influencing the evolution of that trait is nearly impossible. So ideally, you have a system where you have a trait evolving again and again in different organisms, and it's not seemingly under strong selection pressure. But where to find such a trait? Enter earless toads. I'm going to turn on, there is sound. Okay. So, before I go into earless toads, I need to introduce you to the toad ear. So most frogs and toads actually have an ear. You may not have known this, although many of you who know me do. This is a toad, Rhinella spinulosa. You can find it in Peru. And if you pick up most toads, especially here in North America, you're going to find an eye, and if you look right behind that eye, you'll find an ear, shown here by this arrow. And what you can really see from the outside is a thin tympanic membrane, which I'm showing in this 3D model here as this yellow structure. And it's surrounded by this cartilaginous ring for support. That's termed the tympanic annulus, shown in blue. Now, if we zoom inside the skull, you can see that this tympanic membrane is actually connected to the inner ear of the toad, shown here in orange, by this middle ear column made mostly of bones, termed the columella or stapes. And how these structures function is a sound, say from a calling toad or really anything else, will hit this tympanic membrane, and those sound waves will cause the membrane to vibrate. This in turn causes the middle ear column to move in a piston-like fashion, transferring those vibrations from the tympanic membrane to the fluids of the inner ear, where they're sensed by hair cells. This is super similar to how you are all hearing me now with your middle ear. Now, earless toads retain this inner ear and just lack all the middle ear structures, leaving them seemingly no way to get sound from the environment to the inner ear, where it can be sensed. And the crazy thing about especially toads is that earlessness is really abundant. So here I'm showing you the relationships between all toad species for which we know presence or absence of the middle ear. So we have eared species indicated by these orange dots and earless species indicated by these blue dots. These two colors will be used throughout the rest of the talk, so get used to them. And what you can see is that although most toads have an ear, there's more orange than blue. There's a lot of blue in this family and a lot of blue in a lot of different places. So we have a minimum of 12 up to 17 ear transitions in this family alone, which makes toads really unique because in other frog families you only see one or maybe a few losses of the ear at most. And another really fascinating thing about the trajectory of the middle ear in this family is that the most likely scenario is that there was an earless ancestor. So at the beginning of all toads, there was no middle ear. And then there were these two potential subsequent regains of the middle ear. This is the only family of frog or toad where we see regains. One occurred very early in the evolution of this family, giving rise to most eared species and then many subsequent losses of the middle ear. And then a second, more recent regain occurred in a single genus that has mostly earless species and then one small clade of eared species. The other crazy thing is toads are everywhere. Earless toads specifically are everywhere. So here's a world map, and every country that has an earless toad species has a dot, and the size of that dot indicates how many earless species there are in that country. So you can see there's a high concentration of earless toads in Northwest South America, but they're really all over the place. And not only are they geographically everywhere, they're in tons of different environments. So you can find earless toads in deserts. You can find them near raging waterfalls. You can find them in low elevation streams. You can find them in high montane cloud forests. So unlike cave fish or these other convergent systems, we don't see all earless toads in similar habitats, and no one has found a common selection pressure that could explain ear loss in these species. So why are so many toads earless? This is the very basic question that has driven all the research you're about to see. We are not the first to ask this question, but we figured we'd use this really labile toad system to try to address it in a new and comparative way. So I'm going to be asking two main questions in my talk. One is, do earless species share selection pressures that we just aren't seeing? So not environmental selection pressures, but selection pressures in a different way, and I'll get to that in a minute. And second, whether we find evidence of strong developmental bias that could be influencing middle ear evolution. So starting with this first one, wanted to ask whether selection on tympanic hearing might be relaxed. Again, not due to the environment, but instead we actually thought that earless species might be retaining their hearing. And we thought this because frogs and toads, unlike you, if you were to lose your middle ear, are known to have all these extra ways that sound can get from the outside environment to the inner ear. They aren't very well worked out. Here are two potential pathways that people have proposed and tried to test. And to some degree, we know that they are working in some very few species. So we thought maybe that these could be relaxing selection on the ear because perhaps you can retain your hearing without it. So we decided to do a bunch of hearing tests on toads. And we chose toads from this area of the phylogeny indicated by this red bar. We tested 6 eared species and 4 earless, and we were able to capture 3 independent ear loss events. So how do you test the hearing of a toad? You can't ask them to raise their forelimb when they hear a sound. So you can do these really easy, relatively, experiments called auditory brainstem recordings, or ABRs. And what's really nice about them is you can do them right in the field. So you can go out, you can collect a toad, you can put them in this auditory chamber, very high tech that I made. And then you can play tones to those toads through a speaker. And then using electrodes that you put right beneath the skin and over the main nerve that runs from the inner ear to the brain, you can see when the toad hears the sound, when it responds to your signal. And I want to thank Jakob Christensen Dalsgaard. His lab provided the software, and he provided many field seasons of mentorship in Ecuador and Peru, where all these experiments were performed, and kept the electronics running in very humid environments. So the hearing sensitivity is of one species. I'm going to walk you through kind of the data you get from these APRs. So on the x-axis here, you have frequency running from 200 hertz. It's not the best. I'm going to try to break it up. Yeah.
Just sing it, Molly.
That's fine. It's just a really low sound. My mother's biggest worry was a technical failure. Sorry, Mom. Okay. So all the way up to 4,000 hertz, and this is the normal hearing range of a frog or toad. On the y-axis, we have thresholds. This is how loud I had to play the sound before the toad responded. So low on this y-axis, we have really good hearing, and high on this y-axis means it was really poor hearing for that toad. So for this particular species, you see at low frequencies, it can hear relatively well. Here's about the decibel level of normal conversation. It can hear that quite well. But then as you go up in frequency, I'm having to play the sound as loud, about as loud as a lawnmower for them to be able to sense it. So what does this mean for a toad in the wild? Well, this difference in decibel level means if you have hearing up in that high range, and you can hear a sound from 1 meter away, if you were a toad that could hear this decibel range, you could hear that same sound 32 meters away. So that's a big deal if you're trying to hear a predator or prey or a mate. So here are the hearing sensitivity curves of all the species. It's a little bit crazy. But if I can just focus your attention to the left of this line, so at the lower frequencies, you can see that regardless of whether there's an ear or not, you do not see much difference between these curves. And also, they're all sensing sound relatively well in this range. But if you look to the right, that's when it starts to get a little crazy. You see a ton of variation, and it's really at these higher frequencies, everything above 900 hertz, that earless toads are not sensing as well. So we do see a difference between eared and earless toads above 900 hertz. Okay, so they're not completely retaining their hearing. They're retaining low frequency hearing, yes, but not anything above 900 hertz. There's this 10 to 26 decibel difference between the ear and the earless species, which is about a 3 to 20 times difference in distance of being able to hear a sound. So what does this mean? We don't know a lot how toads are using sound to sense predators or prey. There's just not a lot of data on that. But we do know that frogs and toads use sound to find mates, find and attract mates. So we asked what this might mean in our earless species. So now I'm showing you two species, closely related species pair, one-eared, one earless, Rhinella yunga and Rhinella alata. They both call. The eared species calls at this frequency indicated by the speaker, and it can hear that call really well, maybe not surprisingly. And the earless species also calls around 1,000 hertz, and it can hear its call just as well as that eared species. So here's a case in which, at least for social communication, that high frequency hearing class might not be so much of a cost. However, there are plenty of earless species, including outside of the species I tested here, that have much higher frequency calls where they cannot sense those frequencies nearly as well as eared species can sense their calls. So here are two examples in our data set, Rhinella borostandens and Sornifreni guanthamios. They're calling at these really high frequencies, and their hearing is really poor at these frequencies. Okay, so is selection on tympanic hearing relaxed? We say not entirely. There are these high frequency costs, and they seem like they'll impact social communication in at least a few of our species. So we had another idea for selection pressures, which might be universally acting on earless species, and that's that another sense might be enhanced in earless species. And specifically, we wanted to look at vibration, so ask whether earless species were better at sensing vibration than eared species. And we wanted to look at vibration for really two main reasons. One, we know frogs and toads can sense vibration really well, indicating it might be an important sense for those species. And two, the pathway in which vibrations are brought to the inner ear is in direct contact with the middle ear. So there's a number of ways in which removing the middle ear might impact this sensory pathway. So we performed more ABRs. This time, instead of playing pure tones from a speaker, we were able to stimulate the toads with different frequency vibrations using this little platform. And so here I'm going to show you the averages for all our eared and earless species. Again, we have frequency on the x-axis ranging from 200 all the way up to only 900 hertz this time. This is because high frequencies don't travel via vibration very far, so they're not very important. This is why you can hear your roommate's bass really well, but not Adele's high notes. On the y-axis, we have threshold and acceleration. And I want to point out, so again, lower on this graph means you can sense vibrations better. And your iPhone is buzzing somewhat around here. If you are ancient enough to have a 4 especially, that's the exact model of this one. And no matter how extraordinary a human you are, your fingertip can only sense vibration at about this level. So toads are sensing vibration much, much better than you are. So now I'm going to zoom into these lower frequencies so that you can better see differences between eared and earless species. And you can see that the earless species, these blue lines, are trending a little bit lower on this graph. And in fact, we see significant differences between eared and earless species at these lower frequencies, with earless species sensing vibration a little bit better than those eared species. However, I also want to point out there are plenty of eared species on this graph that are sensing vibration just as well as those earless species, so you can have it all. You can retain a middle ear and still have really great vibration sensitivity. So they are on average sensing these low frequencies better. What does that mean? That is a great question that no one has really addressed yet. So we don't actually know how vibrations are being used by frogs and toads to attract mates or predators or prey. What we do know is we have a collaborator working on earless toads, and you can often find them sitting on these leaves. And he'll probe the leaves, causing vibrations, and the toads will try to eat the probe, indicating they might be using it for prey capture. In our lab, we've seen a number of earless species sitting on these ferns, which would be great conduits of vibration. And this picture on the left here is particularly interesting. So Jenny Stanosky, an awesome postdoc in our lab, was injecting this earless species with hormones that ideally make them want to mate with each other. And as soon as she injected them with hormones, they all jumped onto this fern. And she's also seen them calling to one another on ferns. So maybe they're using it for social communication, but it's all very anecdotal right now. We don't actually know. We need more natural history experiments and manipulation experiments to really test that. Okay, so is another sense enhanced? Yes, but again, we don't really know how they're using it, and we don't know, even if they are using it, whether it really compensates for this high frequency hearing cost that we see in our hearing data. So we think that there's really weak evidence that there are these shared selection pressures that can explain this lack of middle ear in so many species, given the sensory cost and its seeming importance. So next we wanted to move on to this idea of developmental bias. So do we see strong evidence of developmental bias in the system? And we have two kind of sub-hypotheses for this as well. The first being whether earlessness might be a byproduct of changes in other skull features. So much like that cavefish example where you lost the eye, but then you gained taste buds, maybe earless species have something else changing and you lose the ear as a result. And really we wanted to look at bony features. So bony features in the skull we thought as particularly important because the precursor cells that ultimately form all the parts of the middle ear also form almost every other bone in the skull. So that's shown by these middle crest streams, and everything that basically has a color on the skull is formed by those same precursor cells. So essentially the ear is developmentally tied to everything. So to get at this question, we performed micro-CT analysis on 55 different species of toad, 39 eared and 16 earless. And we were fortunate that the Smithsonian was willing to just ship me tons and tons of their toad species without much question. And I want to thank Ty Fierro, who is an undergraduate researcher in our lab, who has analyzed or collected all the micro-CT data you're going to see today. And you're going to see even more of it past this. He's done a really great job. So, our overall question is, do we see any differences in shape between these eared and earless skulls? So now I'm going to show you the maximum differences between an eared and earless skull. So first to orient you, we're looking from the top of the skull, and this analysis basically says, what is the absolute, if you could make eared and earless skulls as different as you possibly could, how different are they? So again, we have the trace of an average eared skull here in orange and an average earless skull here in blue. And what you hopefully see up top is they're not crazy different. So in any given dimension, it's changing a hair, a millimeter at most. But I'm going to point out the major axes have changed with these arrows. So these arrows show you how the earless skull is changing in relation to the eared skull. So the earless skulls are a little bit thinner and a smidge longer. And if I show you this other side view of the skull, you can see, again, this difference in length with earless species being a little bit longer. And also there's a slight change in depth, such that earless skulls are slightly shallower. So, do we see these consistent differences in skull shape? When you control for size and phylogeny, you do actually find consistent differences between this average eared and earless skull. However, we think it's unlikely that any of these shifts are causing any developmental issues with the middle ear or causing any sort of trade-off with the middle ear. And also, I want to point out, those were differences in averages, really, but there are earless skulls that we see that are very deep and narrow, and wide and short. So there are plenty of earless skulls that buck those average trends. So next we wanted to move on to this idea that developmental timing might really be affecting middle ear evolution. And we thought timing mattered because the middle ear develops pretty late in a frog or toad's life. So most frogs and toads have this phase where they're completely aquatic and living as a tadpole. And during this phase, if you can see this clear and stained specimen, the skull is mostly cartilaginous, so it's mostly blue. And bone in this cleared and stained specimen is shown in red, and it's mostly just in the spinal column at this stage. Now during metamorphosis, that's that crazy time in a frog or toad's life where they grow all their limbs, they resorb their tail, and they pretty much rearrange everything in their skull. It's during this time that the middle ear just starts to develop. And in many species, it's known that the middle ear doesn't complete development until far into the juvenile stage, so when most other skull features have been fully formed and ossified into bone for a while. So we wanted to ask specifically whether earless species might be developing a bit more slowly than eared species. Because if you're developing the structure late and you slow down your development enough, then you might not get to those structures at the very end. However, comparing development rate in species, especially amphibians, is a little bit crazy. For one, you would have to raise frogs and toads of many species, which one, most people don't know how to do, and two, takes a lot of time and money. And also there's a ton of factors which affect development, so food availability and temperature and all these things that you have to be very careful to control. But lucky for us, there are these indicators of changes in development rate in amphibians. So we know, for example, that if earless species are developing slower, we would expect them to be a little bit smaller in size. So we were able to look at this. We could just go back to our micro CT data. And I looked at overall skull size here, but it trends perfectly with overall body size. And what we see is that a wide range of sizes in the ear column, and earless species are generally small. And so this is a significant difference between eared and earless species, even when controlling for phylogeny. But I want to point out that there are plenty of really small eared species. So it's not as though you can get to some really small size and you can no longer develop an ear. Okay, but we do see this trend in differences in size. Maybe that's an indicator of differences in development rate. But we had some other indicators we wanted to look for. So one, we wanted to know whether they were missing other skull bones, and we really wanted to focus on late forming bones since those were most likely to change. So we went again back to the micro CT data and looked for any bone loss across eared and earless species. And we paid special attention to these 5 bones because they're really late forming in most frogs and toads, and they form right around that middle ear. And what we found was they're not missing any other bones. There was one earless species that was missing 1 bone, the palatines, but overall they're not missing anything else. Okay, so a no-go on that, but we had another idea that was somewhat similar. Maybe they're not completely lacking those bones, but instead just overall ossification was halted in these species, so turning those cartilaginous structures into bone was halted. So we wanted to look at whether overall earless species were less ossified. So again, I'm going to show you a phylogeny of all the relationships between the species we tested. And these barred segments to the right of the phylogeny indicate how ossified the skull is. So the bigger the bar, the more ossified the skull is. And what you can hopefully see is that in our earless species, there was a lot of variation. So there are really unossified skulls, such as this example, where you see not a lot of connectivity between the bones, a lot of gaps. The bone in general is less dense. But we also had earless species with very ossified, even hyperossified skulls. So the skull just looks like a tank. Everything's fused to everything. Everything's really dense. And so maybe it's not surprising then that we found no differences between eared and earless species in this overall ossification measure. Okay, so it wasn't looking great for development rate, but we had one last thing to test, and that is there's this really cool association in amphibians with genome size and development rate, such that if you develop slower, you're more likely to have large genomes. And the mechanism between that is a little hypothetical at this point. Maybe Ken and Rachel will test that one day and tell me the answer. But we know that it's true. So how do you measure genomes in a bunch of eared and earless toads? I did not know until 3 or 4 months ago either. But I went to the Smithsonian and was able to go back into their football field-sized rooms of collections of toads. So just jars of dead things every biologist's dream. And then I was able to sample not just toads, but we looked at eared and earless frogs and toads. And this is just to maximize our sampling of this study. So I extracted heart cells from these specimens and put them on slides, and then I could stain these heart cells for DNA content, and from there we could analyze and approximate genome size across all these species and compare them. Oh, and this was done in collaboration with Marissa Metz, and she rotated as an MCIN student in the Hope Lab and found all these cool differences between ear-to-earless toads that we had not found, and really turned us on to this genome study. So, if you're not sick of them yet another phylogeny of our sampling. And we have ear and earless species. Again, these segments now correspond to size of the genome, such that a larger segment is a larger genome in that species. And although everything looks pretty comparable, when you control for phylogeny, you do find that earless species have larger genomes. Okay, so we have a few indicators that make us think development rate might be important. It might be affecting middle ear evolution. And we have one other line of evidence that was actually known by us when we started this project. And that's that earlier I told you toads were a really weird family. There was way more losses and potential regains of the middle ear than any other frog family. And what we know from the very few species of frog and toad where middle ear has actually been tracked through development is that there are frog species such as Ronopippians, which is a pretty well-studied frog species. And this blue bar here indicates how long past metamorphosis it takes for that middle ear to complete development. So in this species, the ear develops and is complete almost immediately after metamorphosis, hours, at most a few days. There's another frog species that's been looked at, Hyla crucifer, and there's been some other Hyla species that are similar. This species takes 60 days post-metamorphosis, so a couple months to complete middle ear development. And in stark contrast, the only toad that's known that takes up to 1 full year post-metamorphosis to complete the ear. And there's other toad species that we've looked at that indicate that yes, the toads are developing their ear much, much later than these other frog species. So maybe toads are particularly labile with their middle ear because they, one, develop that middle ear very late, potentially much later than their other skull structures. And then on top of that, if you have a shift in development rate, that's going to make that structure particularly easy to lose, and you could lose it without really messing with those other skull features, and that would explain why we don't see any differences in ossification or other bones lost. So that thing I said that would be crazy to try to do, compare development between ear and earless species, we have a postdoc in our lab that's crazy enough to do it, Jenny Stanasi, joined Team Toad, and is actually comparing development rate between two lineages of two species pairs of eared and earless toads. And so growing these up and measuring all sorts of different structures and seeing how development rate differs. And I want to point out that that project and really so much of the data I show today would not exist without these two organizations I have over here. Centro Mbatu is a conservation center for amphibians in Quito, Ecuador, and the Center for Conservation and Development and Sustainability in Oxapampa, Peru. They helped endlessly with collecting these toads. No one in our lab would still call themselves a herpetologist, so we really needed help with that, as well as permits and so many other things. So in sum, earless toads, not hearing as well, sensing vibration a little bit better than eared toads, smaller on average, but despite this difference in skull size, the skulls are approximately similar, not very many changes between them. And we have this difference in genome size, which is really interesting and we think indicates a difference in development rate. So overall, we think that this is pointing towards a developmental bias towards loss of late-forming structures. And that makes earless toads this really sweet system. So you have this convergent trait, this trait that's evolving again and again in different organisms. However, they don't seem to be under any really strong direct selection pressures for losing that middle ear. And in fact, different lineages of earless species might be under different selection pressures, which is really cool. We have this potential underlying developmental bias, which we can really dig into and study the mechanisms of. There's not many systems at all in which we can do that. And before I leave here, as Kim alluded to, I'm going to start new projects and look at the mutations that occur in many different earless lineages and see whether we see shared changes or different changes or selection, especially on genes that pattern the middle here. And I just think that this is a really powerful system, and I'm sure you all agree and want to incorporate it into your lab immediately. So with that, I want to acknowledge so many people. I should check time just to make sure that I can. Okay, so good on time. I want to acknowledge the current iteration of the book lab, which is listed here, with Kim and Jenny highlighted because they are part of what I lovingly refer to as Team Toad. And then the Funk Lab, in all your iterations over the past 6 years, you guys have heard this talk or components of this talk so many times. You've read so many research proposals, so many funding proposals, and I really thank you for your support for the last 6 years. A slew of undergraduates, which I want to acknowledge, gave up personal hours and weekends and summers to 3D model obscure structures of the toad ear, which is my dream job, but I totally recognize not everyone's dream job. My committee, Rachel, Cuba, and Patty, you guys have been great. I've enjoyed every committee meeting, and I'm super excited to talk to you in a few minutes. Collaborators, so many collaborators. I already mentioned Jakob and Marissa, but Juan Wesley, Luis Loma, Lola Guerrero, Alicia Tapia, that's the backbone of the Ecuador contingent of the Earless Toad Project, and we would be nowhere without them. Flor Chama, Juan Carlos Carlo, Amanda Delgado, that's the Peruvian backbone, and really I would have collected all the wrong species and done all the wrong experiments if they were not there. And then I want to thank Nicholas Ditzel and James Schmitz. They helped with the micro-CT portion of this project, and a lot of data that I actually didn't even get to show today. And again, these two organizations, as well as funding resources, as Kim mentioned. This project was mostly funded by an NSF career grant that Kim got a few years ago, Colorado State for funding to go to various conferences throughout my time here, and Sigma Xi for field season funds in the first few years of my dissertation. Okay, now the personal acknowledgement. Thank you, Mom and Dad. There's the obvious reason why I would not be standing here. But there are a million little reasons and ways that you've supported me over the years, professionally and personally, and I really appreciate that. It wasn't all as great as Earless Toads, so I thank you for supporting these other decisions. And then to all of you in Fort Collins over the last 6 years, this is a subset that I could come up with through Facebook and everything else of people, places, and things that I have enjoyed over the last 6 years, dogs and a cat. It's no secret that I love Fort Collins, and a big part of that is the people and the experiences I've had with you all. And so I just really want to say thank you. And if you don't see your face on the slide or aren't sure whether I'm thanking you in particular, come up to me in 3 hours. I'm happy to expand upon this. And I really want to thank the Department of Biology. Dale mentioned this in her talk, but the Department of Biology here is really a stellar place to be a grad student. I think I completely lucked out in coming here. I thought it was a good idea, obviously, but I didn't really understand how great it would be. So I want to thank from those of you that were directly involved and supported my research or my teaching, or just those of you that I saw every 2 years at a random biosocial. Just thank you very much for the community you created here, because I really benefited from it. Now the Hope Lab. I've seen many iterations of the Hope Lab, which started with Kim, and then with Kim and Ava, and then I came in, and now we've expanded to this awesome group of people at the bottom here. And I just want to thank every iteration, especially Ava, my partner in crime for so many years, and hopefully future crimes, definitely future crimes. Also I've enjoyed every iteration though, and I've been so excited to get to know the new members that have recently joined the lab, and I look forward to being in the lab for the next 3 or 4 months. and now Kim. I will be the second, at least second defense where I couldn't, there's just not a really embarrassing photo of Kim. Ava had a pretty good one of Kim eating McDonald's sandwich, which sounds pretty banal unless you know Kim. She's a big foodie. So eating a McDonald's sandwich is about as bad as it gets. But I've thought about this slide for truly 5 years. And not how to embarrass Kim, but just how to thank Kim. I, again, obviously thought it was a good idea to join Kim's lab, but I had no idea the opportunities that she was going to bring me and the support she was going to give me from, you know, just her availability to me, sometimes almost too available. You would send Kim a draft of something and think you could work on at least something else for a few hours, and you'd be back in your inbox in like 30 minutes, which is, you know, great. It's the way I would rather have it. And I've just left the way I describe Kim when I say that I still really like my advisor, even at this juncture, and they usually look confused. And then I just explain it as every time I leave a meeting with Kim, I feel more excited about the research I'm about to go do than when I went into the meeting, no matter my state of mind going in. And so I don't know how that's happening exactly because we're very different people, but I like to think we're rubbing off on each other. In obvious ways, Kim has rubbed off on me, giving me all this advice and making me a better scientist. But the way I think I may have rubbed off on her is that I really like dogs. And Kim is more of a tolerator of dogs. But as this video shows, she's really starting to warm up. So Kim this is Kim bicycling with Cameron Gallenmore's dog when she was watching all the time. Totally IAPIC approved. And you know, the minute here you can see Kim's face, and she looks pretty pleased to be bicycling. And I think that this also, again, shows how awesome our biology department is, because this is what happens when one faculty member goes on vacation and another faculty member watches their dog. Okay, and with that, thank you all very much for being here today. I really appreciate it. And I will take questions.
So you mentioned that you weren't looking at environmental selection pressures that might be shared across these different groups. Is that because other people had already looked at those and ruled them out, or do you think that there could be environmental
I think that there are earless lineages where environmental selection pressures are at play. But I do not think, even if you could adequately study that in all lineages, that it would be some universal pressure. And I think that there are lineages where environment has nothing to do with it. I mean, because you find them calling in just the same environments as earth species, just in puddles on the side of the road. I mean, there just really seems no reason they would not want to hear that. Yeah.
To sort of follow up on that question, do the earless toads, particularly the ones that can't hear well, are they vocal or less vocal, and that might make them less susceptible to predation?
Yeah, so we know that a lot of earless species are calling. People have not looked at this in a ton of earless species. There are earless species that appear to be calling at kind of similar decibel levels and frequencies as eared species. The species that do call, they're definitely trying to attract mates based on their behavior and timing of calling. But there are 2 or 3 species where it's been noted that they call only when really, really close to a potential mate. And so there, even maybe vibration is being more used in the middle here. Or you're just so close, I mean, you're touching them. It doesn't matter. So that could be at play, but basically need one natural issue. Aaron.
Are the eared species
Sorry.
Are there ear species that have stopped calling?
Oh, we know that they used to call. I mean, there are earless species that have never been observed calling. It's hard to say that they definitely don't. And there are earless species that have underdeveloped vocal cords, and sometimes they still try to call, but it's really weak. And all the species around them have calls, so it's a likely scenario for a few species, but it's really rare. But that's one one. Yeah, right. Yeah. Yeah, so, I mean, ideally they're probably using those for other things, right? Sensing other things in their environment, predators, prey, things like this. Yeah. Yes, Grace.
So one of the things you think might be related to earlessness is slow growth rates. So could there be some sort of environmental selection pressures for a slow growth history and slow growth rates that wouldn't be sort of obvious things about the calling environment?
Yeah, I would love to answer this question. I would love for it to take me less than 50 years. There is a trend. Well, it's really anecdotal. We know that there are a lot of earless species that are living in kind of weird habitats. So they're far from water, or they're in these higher elevations where it's kind of cold, and they spend a lot of time seemingly just under rocks. And there's this link between larger genomes, longer development, and kind of what people call weird or extreme habitats. And so I think all of that might be working together. My favorite scenario is that you're in a really weird environment where you don't have pressure to develop fast. So you're not trying to get out of a pond really fast or some other thing. So you don't really need to grow fast. And so therefore you don't, and that relaxes the selection on the genome, which then is kind of allowed to just get really big. That's my favorite scenario, but I have no way to test that in the next minute. Yes.
I have a question about this approach. Correct me if I'm wrong, I'm trying to figure out what you did.
Yeah.
It seems like you did what I would call a systems approach, where you look at sort of random thoughts everywhere.
Mm-hmm.
And if that's what you did, then I'd say, oh, I've done that. I would have picked those that are earless and with ear, closely related. And compare those pairwise, and then see if you get a pattern that holds up all the time. What did you do? Did you do pairwise, or just you pulled everything earless and everything ear?
So depending on the question, we did both. So for the hearing, we designed it such that there were closely related pairs. And I even have a separate pair that I didn't include in that data set for other reasons. So we do have these, as close as we could get, and they are pretty closely related to ear to earless pairs. And we are interpreting the hearing data in that way. The reason I don't have paired comparisons in those ABR analyses is because currently a phylogeny with all those species that's well-founded does not exist. We are creating one, and so even though I do know what their relationships will be, I can't really run the stats yet. And then the micro CT data and all these kind of larger data sets, again, even with the genome study where I was looking across really broad depth, so even in frog species, I chose species that were really closely related, again, as closely related as I could get to do these comparisons. So that when I did those phylogenetic, when I controlled for phylogeny, I was basically doing that, right, teasing apart these closely related species. So both, and ideally species pairs one possible. Yes?
Molly, I think I missed this, but when you did your micro CTs, and you were looking at the ear region, so you showed the skull features, the heart features. But what's in place for the middle ear? What fills the space?
Great question. Soft tissue, so there's one big muscle that if I show you, it almost the middle ear develops right through it. It's right in connection with that tympanic membrane, and the middle ear bone travels right through it, and the middle ear cavity, which I didn't point out, is also there. Basically, that muscle, it doesn't get bigger in earless species. We checked that, but it does just kind of fill in that space a little bit. And really, the area that the middle ear bone and the middle ear cavity take up are really minor. So you just see a little bit more connective tissue in that area, but nothing really cool or different or exciting.
Are those, is that muscle and the middle ear, they share a common set of developmental cells that they originate from?
They don't. If you go deep enough, yes. but probably nothing that makes a lot of sense. But what I think is really interesting and what Jenny is really interested in too is that a lot of these middle ear parts, they grow in coordination with one another, so there's a lot of inductive signaling happening. And because the middle ear and that muscle, which is actually a pretty important muscle, are so close, surely there's a lot of signaling happening between them. So I think there could be something really interesting there, but probably not with the actual precursor cells in the case. Yeah.
So a couple questions both related to correlating genome size. First is that genome size is really tightly correlated to cell size. If you have a smaller body, bigger cell, that means they have a much smaller number of cells. So that way it makes it. And the second one is depending on who you ask and what system you look at, there's some evidence that there is a negative correlation to affect the population size. So maybe there's not relaxed selection for functional reasons, but just inefficient selection for population sizes, failure selection.
Yes. So starting with the cell size. Cell size is actually how we started that study. We looked at cell sizes, actually Marissa looked at cell sizes between our eared and earless species, and we found this really powerful trend that earless species had bigger cells regardless of their body size. However, I do plan to kind of control for body size because I think that's really interesting that if you have much bigger cells and a much smaller body, it's pretty easy to imagine how that would affect your development. So come back to me in a few weeks now that I can stop working on this, and I will have more to say on that. And then the second question about, what was the second bit about? Oh yeah, oh great. Yeah, so when I look at the mutations, I'm going to do a phylogenomics study, and we are hoping part of that will be looking at kind of estimating effective populations or differences between effective population size. Because that's always been a potential, right? If you have something that you can't explain and it's really weird, and also you find these toads in these pocketed populations, it could just be drift. So I'm interested in analyzing that, and that one maybe wait 2 months to ask me. And then hopefully I'll have an adequate answer. Yeah.
I was wondering if, I guess sort of contrary to what Janice was asking, if maybe a selection criteria could be that these environments are really noisy. And so if they have this hypersensitive vibration sense, maybe that's their way of like sending secret codes or something, so they can kind of tune everything else out and focus on those signals in order to do it.
Yes, this has been, this is the story that a lot of people said for a while, especially because there are these really beautiful toads and they live next to waterfalls. And so people said, oh, they don't have ears because they live next to waterfalls and you can't hear anything anyways. And one of my passions on starting this project was that I did not like that story because it was such a story and it just applied to none of these other species. And most species we don't find in habitats that have seemingly really noisy components that would make the ear less important. They could still be using vibration to get around any sort of noise in their environment. But what's also interesting is those earless toads that live next to those waterfalls sit on rocks all day and seem to be doing a lot of their signaling on rocks, where vibration, if anywhere, is least important. So, yeah, there's just not a great story. Doesn't mean it's not out there, but it's not a lot of great evidence out there for environment.
Great, that's it. Let's drink Molly.